There we go. Sorry, I forgot to press the unmute button. We're going to continue our discussion today on muscles. And this afternoon in lab, if you're in the lab, if you're in any of the labs this week, we're going to be taking a look at the nervous system as far as the anatomy of the nervous system. So we'll start this process in the labs this week. Now, a couple things about, registration opened up yesterday. And if you're planning on taking AP2 in my class in the spring, it's listed as a hybrid class. Now, one of the advantages of this is that it can be both on ground and Zoom. In other words, if you're going to take that class, you can take it as a Zoom class just like this one is, or you can come to class and be on the ground. And you don't have to make a decision on which one you're going to go to. If you don't live in the Sevierville area, and obviously you don't want to drive to Sevierville every day, or maybe not at all, you can Zoom, just like we're doing now. If you do want to come to class, you can. So you're going to have the option to choose every day which one you want to do. You're going to get the same lecture either way. Whether you're in the classroom or you're going to be on Zoom, you're going to see the same thing. It's going to get posted to YouTube. It just, you have the flexibility now to choose, to come to class or to attend, by way of Zoom, like you have now. It should be very convenient for everybody to attend this way, and it'll apply to the labs too. So it should be a we're sort of the test case to see if it works, and if it works well, we're going to be able to continue to offer classes that way. So anyway, any questions on anything? Okay, then I'm going to go ahead and go into share screen and go into muscles, and back to where I want to be. Okay. The last thing that we were looking at last Thursday was the sliding filament model of contraction. Now, I said the sarcomere is the contracting unit of skeletal muscle. So in a muscle cell, there are probably billions of sarcomeres. The sarcomere is the structure that goes from Z line to Z line. As we know that we have light and dark bands. The light bands are made up of actin, the thin filaments. The dark bands are made up mostly of myosin with some overlap of the actin. The actins extend from the Z line to about the middle of the myosin. So the Z line is the middle of the I band. The I band is the I band is the light band, and the A band is the dark band. In the middle of the I band, we have a Z line. The Z line is protein that attaches, that the actins attach to. So every sarcomere goes from a Z line to a Z line. This is what it looks like. Let me go back. Let me go back one slide here. This is what a sarcomere looks like. Light band right here on the left-hand side, light band on the right-hand side, dark band in the middle of the light band. That's the Z line here. To the right of this line is a sarcomere. To the left of this line is another sarcomere. Over here at the other side, you have to the left of the line is this sarcomere, to the right of the line is another sarcomere. So these sarcomeres are lined up one after another, after another, after another in every myofibril. So you could have billions of these sarcomeres in every cell. But the structure always stays the same. There's a Z line in the middle of the I band on each end. There's an A band in the middle. The A band is the dark band. The I band is the light band. So we have a light band on either end of a dark band. The middle of the light band to the middle of the next light band is the sarcomere. At the point of contraction, the thin filaments are going to slide over top of the thick filaments. Now, the actins, which make up the thin filaments, and the myosins, which make up the thick filaments, don't change their length. They just slide over each other. That's all they do. The only thing that changes its length is the distance of the sarcomere from Z line to Z line. The sarcomere shortens. The Z lines get closer together. That's all that happens. That's enough. So the Z lines are pulled in as the actins are slid over top of the myosins. The Z lines are pulled in on either side. It looks like this. We see here that our the Z lines are much closer together than they were before. The I band has shrunk, and so the sarcomere has contracted. We see that we pulled, the adjacent sarcomeres are pulled closer to each other. Now, this is happening in every sarcomere in the myofibril, with the rods within, all those rods within the muscle cell itself. And they're all doing it at exactly the same time with exactly the same amount of strength. A muscle cell will contract fully every time it's stimulated. There's no such thing as a partial contraction of a cell. Every cell that's stimulated will contract fully.
But I told her.
And this is what it looks like, as far as the animation goes. See, the Z lines are being pulled in as the actins are being pulled across the top and the bottom, if you will, of the actins, of the myosins. So the Z lines move closer together. That's the only thing that shortens up. The actins and myosins don't shorten. So how do we get to this point? How do we get the muscle cell to contract? We know that the sarcomere is shortening. That's, we know that the sarcomere shortens and the myofibrils shorten and the entire muscle cell shortens. Muscle fiber and muscle cell, again, fiber and cell mean the same thing. So this is under nervous system control. We have what are known as the somatic motor nerve. This is a nerve that leaves the brain and goes out to the individual muscle cells. Every muscle cell is in contact with a nerve. So every muscle cell is controlled by a motor nerve. And the motor nerve sends a nerve impulse down to that cell and causes it to contract. Now, the excitable cells can do this. A nerve cell can generate a nerve impulse. That nerve impulse can be passed down through the nerve to a muscle cell, and that nerve impulse will stimulate the muscle cell to contract. Now, we call this an action potential. The term nerve impulse is fine, but the proper term here that we use much more frequently is called the action potential, because it's based on charge on the membrane of the muscle cell. Well, it's actually based on the charge on the membrane of the nerve cell, and that charge being passed to the muscle cell. Because the nerve cell has a charge on its surface, the muscle cell has a charge on its surface. And if we change that charge, if we change the charge, we generate the nerve impulse, which we call the action potential. It's easier to say action potential in the muscle cell and action potential in the nerve cell than to say nerve impulse. And to make this work, we have to use a chemical. We have to use a chemical known as a neurotransmitter. Neurotransmitter gets released by the nerve cell and allows the impulse, the action potential, to essentially jump from the motor nerve to the muscle cell, because the motor nerve that stimulates the muscle cell is not hardwired. It's not physically attached to the muscle cell. There is no direct connection. It is an indirect connection. There is no contact. There's no physical contact between the nerve and the muscle cell. So we use a neurotransmitter to pass the signal from the nerve to the muscle. And the neurotransmitter that we use in skeletal muscle is called acetylcholine. It is the most well-known and most studied of the neurotransmitters in our body. We have about 60 different neurotransmitters that we use all the time. And in skeletal muscle, we consider that acetylcholine is an excitatory neurotransmitter. It causes it allows the muscle cell to contract. Lots of neurotransmitters are inhibitory. They stop actions or slow things down. But in skeletal muscle, we see acetylcholine as a stimulatory or excitatory neurotransmitter. Now, all of our cells have a charge on them. All of our cells in our bodies have a charge. It has to do with the concentration of sodium and potassium. And this is where sodium and potassium are very, very important to us. There is a charge across the membrane, and it's caused by the presence of sodium ions and potassium ions. Now, sodium tends to be present on the outside of every cell of our bodies in high concentrations. Sodium's in plasma. Plasma becomes interstitial fluid. Interstitial fluid surrounds every cell. So we've got lots of sodium outside of all of our cells. Inside our cells, we've got potassium. Not as much potassium as we do sodium on the outside. So we have sodium on the outside and potassium on the inside. Well, high levels of sodium like to diffuse across the membrane. We know that. And potassium on the inside likes to diffuse out because there's more sodium outside than there is inside the cell. And there's more potassium inside than there is outside. So they try to leak out or leak in. And we have to use our sodium potassium pump. There it is, our sodium potassium pump, to try and control this. We don't want sodium leaking in. We don't want potassium leaking out. Now, if you have all these sodiums outside the cell, the outside of the cell tends to have a positive charge. So what happens though if you have less potassiums inside? If the number of potassium ions inside is less than the number of potassium, if the number of potassium ions inside the cell is less than the number of sodiums outside, counting them up 1 to 1, you've got 3 times as many or 4 times as many, or however many you've got, more sodiums outside the cell than potassiums inside. So there's more positive charges outside than there are inside. That creates a charge. We call that a membrane potential. And we create a charge. And we say that the outside of a cell is positive because all these sodiums outside the cell. And the inside of the cell is negative with respect to the outside. Yes, I know that you've got potassiums on the inside of the cell, and the potassiums are a positively charged ion. They're a cation, just like sodium is, but there's fewer potassiums than there are sodiums. So there's a lot more positive charges outside than there are inside. So we say the inside of the cell is negative with respect to the outside. Take it at face value that the outside's positive, the inside's negative. And that creates what we call a resting membrane potential. There's a charge now that has formed on the membrane of the cell, and they're different. Different cells have different charges. So with the presence of sodium outside and potassium inside, and we're always pumping sodiums back out and always pumping potassiums back in because we want to keep that difference. We want to keep it high sodium outside and somewhat high potassium on the inside. And that keeps a positive charge outside and a negative charge inside the cell. And we are polarized. We have a positive outside, a negative inside. And since you have, yes, we have less positive charges inside than we do outside. So with respect to the outside, we are negative. Face value, positive outside, negative inside. The charge on different cells depends. On muscle cells, the charge is across the membrane of a muscle cell is minus 90 millivolts. That's the difference in the charge from the positive outside and the negative, if you will, inside, minus 90 millivolts. Nerve cells are minus 70 millivolts. And we say that they're polarized just like a battery with a positive side and a negative side. Batteries have a positive end and a negative end. The flat end is the negative end, the bumpy end is the positive end. So, and we say that the membrane potential, the V sub M, the membrane potential, I don't know why it's called V sub M, but that's what we call it. The membrane potential for a muscle cell is minus 90 millivolts. And we can change that. We can change that and generate an action potential, an impulse, if you will. Now, to control this, we have gates and channels in our membrane. We have what are known as gated ion channels. Now, we know that there are different channels in the cell membrane. There's, aquaporins that allow water to come through. We have just channel membrane, channel proteins that are just essentially tubes, for sodium and potassium and other things. We have carrier proteins that are a little more specific. We have carrier proteins that function as the sodium potassium pump. They require energy. So we have very specialized channels. Now, the most specialized channel has a gate on it, an actual physical gate that will open or close the channel. And you see sort of something like that with the sodium potassium pump, the gate. There is no gate in the sense, but the sodium potassium pump changes its shape to open and shut. Well, with a gated channel, there's an actual physical gate that opens and closes. Now, there are two types of gates, and they will open or close to either let an ion go through or to stop an ion from going through. And the gates are specific. There's a sodium gate, there's a potassium gate, there's a sodium channel, there's a potassium channel, there's a calcium channel, there's probably a phosphorus channel too. And these channels are specific for the ions and they are gated. And the gates are controlled either by a chemical that lands on them to open them up or close them. We call it a ligand gate or a chemical gate. I don't know why we call it ligand, but we call it a ligand gate or a chemical gate. And this is one where we use the neurotransmitter. The neurotransmitter is the chemical that lands on the ligand gate and causes it to open up or causes it to shut. Because neurotransmitters can be excitatory or inhibitory. We also have the voltage gate. This gate will open or shut depending on the change in the charge on the cell membrane. Because we know that right now at rest, it's minus 90 millivolts, but it can change. And that voltage gate can open or shut. It's like a garage door opener. And the garage door is shut most of the time. When you press the button on your garage door opener, the garage door opens up. But it only opens up for your signal. And you don't press a button and see a half a dozen garage doors go up around the neighborhood. You only have your door going up. Now, so you have a voltage gate that opens up when there's a change in the charge on the membrane. It has to be a specific change in the potential on the membrane. Potential means charge. So we use both of these types of gates when we have a muscle contraction. We also use these same gates, same type of gates, the way our nerves function too. So we're going to talk about this again when we get into chapter 11. Now we know this, okay, so we have two types of channels in our membranes, and all cells have channels in here. Leakage channels, always open. Aquaporins, carrier proteins, channel proteins, everything comes in, no restrictions. That's what we have here happening at the example on the top. Here comes sodium leaking in through one of these leakage channels. No controls on it. But a voltage channel or a ligand channel, a chemical channel, are restricted. Here's a gate right here. This is a sodium channel here, but it is either a voltage gate or a chemical gate. And it will only open up if the voltage allows it to, or the neurotransmitter allows it to. And once the gate opens, then sodium can come in. When the gate's closed, sodium can't come in, or potassium can't come in or go out. So the gated channels are controlled, either chemically or electrically. A voltage gated channel for potassium, for example, may allow potassium to leave the cell. Now, normally, here we have a gate on the channel for potassium. Potassium's not leaving the cell, but if we change the charge at the gate, there goes the potassium. Potassium can leave. If we use a neurotransmitter, we have a receptor site on the gate right here. And that neurotransmitter has to be specific for that receptor site. It has to function like a key into a lock. It only works if the right key is put in the lock, because if it's the wrong key, nothing happens. And remember, we have 60 different neurotransmitters, so we're not going to have 60 different neurotransmitters trying the lock to see what works. Only the one that works will come in, land on the receptor site, cause the gate to open up, and it'll allow potassium, in this case, allow potassium to leave the cell or to come in the cell. It depends on what the gate's going to do and what the neurotransmitter is going to do. But a ligand-gated channel always requires a neurotransmitter. Ligand-gated channels always require a neurotransmitter. Electrical gates always require a change in the charge on the membrane. So let's park that aside for a second here and consider how we're going to get our muscle cell to contract. We're going to use those gates here. So for a muscle cell to contract, muscle fiber, muscle cell, there's 4 steps. Our motor nerve has to stimulate the muscle cell. Now, every muscle cell is in contact with a motor nerve. There is a nerve ending indirectly connected to every muscle cell. So every muscle cell is under nerve control. So we have to stimulate the muscle cell by the motor nerve. We call it a motor nerve because that's what it does. It is a somatic motor nerve signal coming out of the brain saying, raise your right arm, lower your right arm, whatever. we have for a muscle cell to contract, we have to generate an action potential, a charge in the cell membrane of the muscle cell. We have an action potential coming down the motor nerve. We have to reproduce that charge in the muscle cell. We have to send that charge across the cell membrane and down into the cytoplasm of the cell. And then we have that charge actually triggers the release of calcium from the sarcoplasmic reticulum. The reason that we have an action potential in the muscle cell is so that action potential, that electrical charge, will open up the terminal cisterns on either end of the sarcoplasmic reticulum surrounding every sarcomere and cause calcium to flood out into the sarcoplasm, into the cytoplasm of the muscle cell. Because calcium is key here. So these are the four things that have to happen to trigger the muscle cell to contract. Now, step 1 and step 2 take place at a structure known as the neuromuscular junction. It is the indirect connection between the motor nerve and the muscle cell. There's a gap there. The gap is called the synaptic cleft. You probably heard the term synapse. The synapse is where a nerve connects to another nerve, connects to a muscle, connects to a gland, but they're all indirect. There's always a gap in between here. And so the neuromuscular junction is where the motor nerve indirectly connects to the muscle cell. So the stimulation by the nerve and the action potential that gets developed, created in the cell take place at the neuromuscular junction. The last two steps occur when we propagate the action potential down across and through the cell membrane, down the T-tubules to every terminal cistern of every sarcoplasmic reticulum, surrounding every myofibril, surrounding every sarcomere, so that the whole area floods with calcium, and that allows us that triggers the myosin heads to grab onto the actins. We call it excitation, contraction, coupling. So here we see the motor nerve right there, and where it indirectly connects to the muscle cell is the neuromuscular junction. There is a structure on the muscle cell called the motor end plate. There is only one connection per muscle cell. There's only one motor end plate on every muscle cell. So there's only one connection there. And the axon terminal connects there at the neuromuscular junction. Now the motor nerve extends its cytoplasm and cell membrane from the cell body all the way out to the muscle cell through a structure known as the axon. The axon is the structure of a neuron that the neuron uses to send its action potential. Now the axon can be long, very, very long, thin structure. You know, the sciatic nerve in our leg, the sciatic nerve, the cell bodies of the cell body of the neuron is up somewhere near the spinal cord, but the axon extends all the way down to our big toe. And so the sciatic nerve consists of thousands of these axons linked side by side by side. The axon ends in a structure known as the axon terminal. And the axon terminal is what forms half of the neuromuscular junction. The axon terminal rests slightly above the motor end plate. So every muscle cell will have an axon terminal indirectly in contact with it. So every muscle cell has one neuromuscular junction with one axon terminal from a motor neuron on it. It's only one, it's one nerve ending, one muscle cell. One nerve ending, one muscle cell. The axon can separate into up to 300 terminals. So one motor nerve can stimulate up to 300 muscle cells. However, we see later on that the fewer cells that you're stimulating, the better control you have. So anyway, let's move on here. So the axon terminal is the end of the axon. There is a gap at the neuromuscular junction. It's called the synaptic cleft. It's filled with a fluid, sort of like a gel in there. And at the end of the axon terminal are vesicles that contain the neurotransmitter, acetylcholine. In the motor neuron, the neurotransmitter is going to be acetylcholine, and it's going to be stored inside these little vesicles. It's going to be made and stored in the vesicles at the end of the axon terminal. And when the action potential in the motor nerve comes down the nerve and hits the axon terminal, it will release the acetylcholine into the synaptic cleft, and it will drift across to the motor end plate on the muscle. And at the motor end plate, there are acetylcholine receptors. And it looks something like this. Here's the axon up here. Here's the axon terminal here. This little, it's a bulb, okay? And inside the axon terminal, the end of the axon, you have vesicles with acetylcholine. Now, this space in here is called the synaptic cleft. Now, there's only one motor end plate and one neuromuscular junction per cell. Now, the cell membrane has all these folds here to increase the surface area, and the axon terminal will release acetylcholine into the synaptic cleft at the motor end plate. This whole thing is the neuromuscular junction. It's not a direct connection. They're not hardwired. They're indirectly connected. And so we see these little yellow dots here represent the acetylcholine that is being released into the synaptic cleft, allowing it to drift over to the motor end plate. So have we got a contraction yet? No, not yet, but we're getting close. So the action potential in the nerve comes down to the axon terminal. Nerves generate action potentials. That's what we call the nerve impulse. The action potential comes down the axon terminal. When it hits the axon terminal, there are voltage-gated channels for calcium present at the axon terminal. The action potential causes them to open up and allow calcium to come into the terminal bud, the bulb, whatever, if you will. The presence of calcium there causes the vesicles containing acetylcholine to fuse with the cell membrane of the axon terminal and release the acetylcholine into the synaptic cleft. Let me back here. That's what's happening here. These little purple things here are the voltage gates for calcium. When an action potential comes down here through the axon to the axon terminal, the charge on the membrane causes this voltage gate to open up and calcium comes in. That gate is controlled electrically. And so an action potential is a wave of energy moving down the nerve, causes this gate to open. Calcium comes in, floods the area, causes these vesicles to fuse with the end of the axon terminal, releasing acetylcholine into the synaptic cleft. The acetylcholine starts drifting across to the motor end plate. Okay. There are acetylcholine receptors on the motor end plate. Those receptors are located on chemical gates for sodium. on the cell membrane, on the sarcolemma of the cell membrane at the motor end plate, sodium gates, ligand gates. The chemical that opens up those ligand gates, those chemical gates for sodium, is acetylcholine. Acetylcholine binds to those gates, they open up, and acetyl and sodium goes into the muscle cell. Sodium, which was outside the cell, sodium is always outside our cells. When we open up the sodium gates at the synaptic cleft, the sodium rushes in to the cell. That's what it does. It's just, it's always in high, sodium's always in high concentration outside our cells. It's always trying to get in. Acetylcholine comes across the synaptic cleft, lands on the sodium gates. The sodium gates open up, sodium goes into the motor end plate region. Let me see that back here. These little purple things down here are sodium gates. They're not color coordinated or anything. Each one of these sodium gates has a receptor site for acetylcholine. When acetylcholine lands in these gates open up, and sodium, which is always present outside the cell, will start moving into the cell, the muscle cell at the motor end plate. So now the outside of the cell, which was positive, and the inside of the cell, which is negative, is suddenly getting an influx, a rush of sodium coming in here, a bunch of positive charges coming in here, disrupting everything. And then the leftover acetylcholine is then broken down by an enzyme known as cholinesterase, acetylcholinesterase, or just cholinesterase, because we can't leave the acetylcholine there because it would keep the gates open all the time. We don't want it there. We don't want the gates to stay open all the time. We want to get rid of the acetylcholine so we can do this again. Now, problems with acetylcholine. Acetylcholine is absolutely necessary for muscle contractions, but there's lots of things that can interfere with acetylcholine. There is an autoimmune disorder called myasthenia gravis, and you see the eyelid characterized by drooping eyelids. Drooping eyelids, the upper eyelids droop. You see here in the picture here, it's drooping down here, covering the eye. There may be problems swallowing, there may be problems talking, and there is an overall muscle weakness because in myasthenia gravis, your own immune system attacks the receptor sites for acetylcholine. It destroys the acetylcholine receptor sites on the sodium at the sodium gates, and so we don't get as much sodium coming through. We can't generate an action potential in the motor nerve, in the muscle cell, because that's what happens when sodium comes in. It generates the action potential in the muscle cell, but without enough sodium coming in, we don't get that. And if we don't have the gates functioning, we're not going to get the sodium. And so we are attacking our own acetylcholine receptors at the motor end plates of all this skeletal muscle. So we have our own autoimmune disorder attacking it. There are chemicals which destroy the enzyme that breaks down acetylcholine. Many insecticides are cholinesterase inhibitors. They prevent the enzyme from breaking down acetylcholine. And in insects, it kills the insect because the insect muscles contract and can't relax because they're constantly being, the sodium gates in the insect are being held open because there's still acetylcholine there. We don't break it down. And it does the same thing to people, even though we're not insects. If we're exposed to a pesticide that blocks the cholinesterase enzyme from breaking down acetylcholine, we get muscle contractions that don't let go. You don't like that either, so it can kill us. Anyway, myasthenia gravis, whatever, likely an autoimmune disorder. caused by the destruction of the acetylcholine receptors at the motor end plate. So now this, hopefully you can hear this, but let's go ahead and watch this.
Nerve impulses, also known as action.
Okay, so whether you could hear it or not, the animation is very good, but I apologize if you couldn't hear it. But what we're seeing here is the action potential comes down the motor nerve, triggers the release of acetylcholine, which drifts across to the motor end plate, opening up sodium channels. The acetylcholine is the chemical messenger. It lands on the ligand gate for sodium, opens it up, and sodium, which is always present outside the cell, rushes into the motor end plate. Now, all those sodiums have positive charges. They change the charge on the membrane at the motor end plate. It's a localized change. So we see a change in the charge at the motor end plate. So let's go through this. The sarcolemma we know is polarized, positive on the outside, negative on the inside, negative with respect to the outside. The action potential that forms, the nerve impulse, if you will, the action potential is caused by a change in the charge on the cell membrane of the muscle cell. We have to have a change. We know the motor end plate is at minus 90 millivolts. We want to change that. But then we got to bring it back to what it was. The terms here are depolarization and repolarization. Remember, it's polarized. It's positive on the outside and negative on the inside. So the excitation part occurs when we have at the motor end plate, we have chemically gated channels. They are ligand channels for acetylcholine. On either side of the motor end plate are voltage gated channels. Now what will happen is at the motor end plate, acetylcholine drifts across, lands on the ligand gated channels for sodium, and they will open up and sodium will come in at the motor end plate and start changing the charge on the membrane. It's normally at minus 90 millivolts, but we will see an increase in the charge from minus 90 millivolts. It will become less negative as more and more sodiums come in. Remember, sodiums are positively charged. As the sodiums come in, the charge at the motor end plate changes from minus 90 to minus 80 to minus 70 to minus 60 up to minus 50 if it gets there. There's no guarantee it's going to hit minus 50, but if it does hit minus 50 is the threshold level where the voltage gates will open up. Now the voltage gates are on either side of the motor end plate. They will only open at minus 50 millivolts or above. They won't open at minus 60 or minus 55 or minus 51. They'll open at minus 50 because we've gone from minus 90 to minus 50. As more positive sodiums rush into the motor end plate, we depolarize. We change the charge. We make the charge on the motor end plate less negative. We go from minus 90 to minus 50. If we hit threshold, which is minus 50, then the voltage gates on either side of the motor end plate will open up. And those are voltage gates for sodium. They only open if enough sodium comes in at the motor end plate. If we don't hit minus 50, then nothing happens. There is no, we don't reach threshold, nothing happens. There's no muscle contraction if we don't hit threshold. Gotta hit threshold. Nothing happens if we don't. No muscle contraction. Now, if we do hit threshold, if we are fortunate enough to hit threshold where we change the charge in the membrane at the motor end plate from minus 90 up to minus 50, that will trigger the opening of sodium channels on either side of the motor end plate. Sodium channels open up and allow even more sodium to rush into the cell membrane, rush through the cell membrane into the sarcoplasm of the muscle cell. More sodium comes in. More positive charges come into the cell. Right now, at the motor end plate, all we've done is have sodium come in and have a localized event taking place at the motor end plate. We may or may not get the threshold value. We may or may not hit minus 50. If we don't hit minus 50, then nothing happens. We didn't hit threshold, everything stops. If we do hit threshold, then the voltage gates on either side of the motor end plate will open up and allow more sodium to rush in. And that will continue the process. That will continue the process of letting sodium rush in, and we'll see a localized, what we call a depolarization event on each side of the motor end plate. We'll see a depolarization event occurring at the voltage gates on either side of the motor end plate. So here's our axon terminal right here. Calcium comes in to trigger the release of the neurotransmitter. The neurotransmitter vesicles fuse with the membrane of the axon terminal. They get released into the synaptic cleft. They land on the sodium gates down here that are ligand controlled, chemically controlled gates. They open up and we create a potential. We depolarize locally. Depolarize means we're changing the charge. We are reversing the charge. We're polarized at -90 and we are reversing it from -90 to -80 to -70 and so on down to maybe we hit threshold, maybe we don't. While this is going on, this is a localized depolarization event that spreads, that will spread to the voltage gates on either side. This is a voltage gate here. And there's another set over here. There's more over here. These voltage gates, if we hit threshold at the motor end plate, the voltage gates adjacent to the motor end plate will open up. And that will cause a depolarization of the cell membrane. We will change the charge. We now have an action potential forming here. Now, once depolarization starts here outside of the motor end plate, it won't stop until it's done. Because now we're going to change the charge from -90 to -50 to open up these gates. And sodium rushes in and it will continue to change the charge on all the voltage gates spread out through the length of the muscle cell. On the surface of the muscle cell are all these voltage gates in place. And as one sodium gate opens up and causes a depolarization event, that will change the charge in the gate next to it, and it will open up and allow sodium to come in, and the next one will open up and allow sodium to come in, and the next one after that will allow sodium to come in, and we will see an overall change in the charge of the cell membrane from -90 all the way up to +30. So we have our depolarization event starting here. Our action potential in the muscle cell, which is what we're trying to generate, occurs right here when the first voltage gate reaches threshold. When the first voltage gate reaches -50, because we've depolarized the area around the motor end plate. If we hit -50, this gate opens. And from then on, the action potential will run across the membrane until it completely runs through the membrane. Once it starts, it doesn't stop till it's finished. It opens up the sodium channels. Sodium rushes in. We depolarize this region. That region goes to from -90 up to threshold. It opens up the gate next to it and the gate next to it, and more sodium rushes in. We continue to change the charge on the membrane. Now, when we get to about 0, we've gone from -90 up to 0 millivolts. Somewhere along in that neighborhood, we close the sodium gates and open up the potassium gates. So now to summarize this without pictures, once we release the acetylcholine into the synaptic cleft, it binds on the sodium ligand gates on the motor end plate. That causes a localized depolarization. Sodium rushes in. All these positive charges are coming into the motor end plate of the muscle cell. The charge is changing as more sodiums rush in. Sodiums are rushing in because there's always sodium outside the cell. Sodium is rushing in. The charge is changing from -90 to -80 because they're all positive charges, so we're reducing the negative charge. The inside of the cell becomes less and less negative. It's a localized event. We create a potential, an action potential, where we create what's known as the end plate potential. We haven't made an action potential yet because we may or may not hit threshold. But if we hit threshold, -50, then the voltage gated channels on either side of the motor end plate will open up. and that will trigger a massive influx of sodium. That massive influx of sodium causes a depolarization wave which spreads across the muscle cell. Because once you depolarize a voltage gate, it allows so much sodium to come in that it will depolarize the region next to it, and the one after that, and the one after that. And every gate that opens up will allow more sodium to come in. And the action potential starts and it will not stop until it runs through the entire cell. And the action potential goes across the cell membrane, the sarcolemma, from one voltage gate to the next one, causing that area to depolarize. Each gate opens up as it hits threshold. And when it opens up at threshold, it will allow more sodium to come in, which opens up the gate next to it, and that will depolarize that region and open up the gate next to it, and it spreads rapidly across the entire cell membrane. But we can't stay depolarized. As we go, our charge goes from -90 up to +30. We can't stay like that. So somewhere between 0 and +30, the sodium gates shut. Remember, they're voltage gates. They open at -50 and they close around somewhere around 0 to +30. At that point, the potassium gates, the voltage gates for potassium, open up. And now potassium starts leaving the cell. Sodium has rushed in, potassium leaves. This is the repolarization event. Because what we're trying to do is reestablish the charge on the membrane. Because we've gone from positive on the outside and negative on the inside to suddenly positive on the inside and negative on the outside, because all the sodiums came in. And there were suddenly more sodiums inside than there were outside. So the charge in the membrane went from, when it depolarized, it flipped from positive outside to negative outside, from negative inside to positive inside. So we open up the sodium, we close the sodium gates, open up the potassium leaves the inside of the cell and goes outside and changes the outside of the cell from negative back to positive. It doesn't matter that there's potassium outside now and sodium inside. We have reestablished the positive charge on the outside. Sodium potassium pump will fix everything very, very rapidly after repolarization takes place. After we get our positive charge back outside, the sodium potassium pump will very quickly move the ions back into their proper position. But we now have to repolarize the muscle cell so we can do the whole thing again. Depolarization, once the action potential starts, doesn't stop. It's an all or none proposition. You hit threshold, you're going to have an action potential that runs across the muscle cell. It's going to go all the way through the cell and down through every T-tubule to every terminal cistern. And it won't stop. Now, whether it starts is another matter, because the only way it will hit threshold is if we get enough sodium to come through the ligand gates. Did we open up enough sodium channels? Did we have enough acetylcholine to open up those channels? And if we did, great. Then we're going to have, if we hit threshold, then we're off and running. We will get an action potential that spreads across, a charge that spreads across the muscle cell and will not stop until it's done. But it depends on the number of channels you have, the number of ligand channels you have, the amount of acetylcholine that you open up. So now, depolarization and repolarization. Don't get these confused. When sodium comes in to a cell, it becomes depolarized because sodium is positively charged. The outside of the cell is positive, the inside of the cell is negative. Depolarization occurs when sodium comes in and changes the charge. So if the outside of the cell is positive, the inside is negative, sodium comes in, and now the inside of the cell is positive and the outside is negative. Repolarization is when potassium goes out to reestablish the positive charge on the outside. So here we see depolarization, sodium's coming in, repolarization, potassium goes out. We have these voltage-gated channels for sodium and potassium that open up and close to allow us to change the charge, to depolarize the cell, but we got to repolarize it to get us ready to do it again. So how long does this all take? Not very long. Somewhere between 5 and 10 milliseconds, a thousandth of a second. So we're not even aware of it. So the change in the charge is shown here on this graph. The resting potential is minus 90 millivolts. The sodium channel is open. We hit threshold at minus 50, which is right around in here. We depolarize all the way up to plus 30. At plus 30 millivolts, the sodium channels close and the potassium channels open. We start repolarizing, and we repolarize ourselves back down to minus 90 millivolts. Less than 10 milliseconds for this to occur. Less than 10 milliseconds for this whole thing to occur. So our resting potential is minus 50. I'm sorry, the resting potential is minus 90. If we can reach threshold at the motor end plate of minus 50, then we'll trigger the voltage channels on either side of the motor end plate to open up and have a massive influx of sodium. And we'll see this depolarization event occurring, bringing our charge in the membrane up to plus 30 millivolts. But that shuts down the sodium channels and it opens up the potassium channels. And potassium now leaves the cell and reestablishes the positive charge on the outside. And that will continue until we get back down to minus 90, and then the potassium channels close. So the action potential. This summarizes, shows the whole thing here. Our resting potential is minus 90. Important number to remember. Threshold is minus 50. Depolarization goes from minus 90 to plus 30. Repolarization takes us back from plus 30 to minus 90. Sodium depolarization is sodium coming in. Repolarization is potassium going out. And we go all the way up to plus 30 and we come back down to minus 90. So that's all well and good, but how does that get us to a muscle contraction? You know, we haven't got the contraction yet. We've been sending action potentials across the muscle cell. We've been seeing the movement of sodium and potassium, you know, in 5 milliseconds. But how does that get us a contraction? The action potential moves down the T tubules that are embedded in the cell membrane. The T tubules are the extensions of the cell membrane, and they run down through every individual myofibril. There's a T tubule that surrounds every myofibril at the ends of every sarcomere. The action potential comes down the T tubules and opens up calcium channels at the terminal cisterns of the sarcoplasmic reticulum on each sarcomere, each contracting unit. And that floods the entire cytoplasm of the cell. All the myofibrils within the cell are flooded with calcium because they all release, all the sarcomeres are flooded with calcium at exactly the same time because every one of the terminal cisterns at the ends of every sarcomere open up, and all that calcium floods out into the cytoplasm. And that will lead to the contraction. Now our action potential is done. The whole purpose of the action potential, the whole purpose of everything up to this point is to cause the release of the calcium. That's what we're trying to get, is to try to get the calcium to come out of the sarcoplasmic reticulum. Everything we've done so far, the motor nerve, the acetylcholine, the synaptic vessels, the motor end plate, the depolarization of the motor end plate, the depolarization of hitting the threshold at the voltage gates on either side of the motor end plate, the rushing of sodium, the depolarization to plus 30 and the repolarization back down to minus 90. All that's done to generate the action potential, the charge that will go down the T tubules and open up the terminal cisterns and release all this calcium. In fact, the action potential is over before any of this happens, because all we've done is we've sent the charge down here through the T tubules, and every terminal cistern at the end of every sarcomere opens up. And this leads to contraction. Let's see if this works. Or T tubules. Okay, so how does calcium do that? Within the sarcomere, you've got the thin filaments and the thick filaments. On the thin filaments, you have tropomyosin blocking the binding sites for the myosin. You have tropomyosin, you have troponin stuck in the middle of all this. Now, with tropomyosin blocking the binding sites, the myosin can't get there. The myosin can't grab onto the actin. Our muscle can't contract. The muscle cell can't contract because the myosins are all blocked. The T tubules release calcium that floods the sarcomere. Every sarcomere is flooded with calcium. Calcium binds to troponin. And when it binds to troponin, it causes the troponin to roll over. It functionally rolls over. And when it rolls over, it drags the tropomyosin with it because the troponin is attached to the tropomyosin, and it drags the tropomyosin over with it, and that exposes the binding sites. And the myosin heads can grab onto the actin And then they will, when they grab onto the actin, the myosin heads will pivot and pull the actins towards the middle of the sarcomere. And then if there's ATP present, they'll let go and recock and do it again and continue to pull towards the middle of the sarcomere. So everything we've done up to this point is done to release the calcium out of the sarcoplasmic reticulum so it can bind to the troponin, causing it to roll over, causing the tropomyosin to roll over, exposing binding sites that the myosin can now attach to the actin and give it a good yank towards the middle of the sarcomere. And this is happening in every sarcomere, in every myofibril of the muscle cell. And every cell is contracting to its full, every myofibril is contracting because every sarcomere is contracting. And the muscle cell, the individual cell, contracts to its fullest extent when it does this. So everything was done just to get this calcium released that allows the binding sites to be exposed. And they will stay exposed as long as we are stimulating the muscle cell. Once the stimulation stops, then the calcium goes back to the sarcoplasmic reticulum, the troponin rolls back over and the tropomyosin rolls over and the binding sites are blocked. And this is a great place to stop. And we will continue with this on Thursday. So everything we've done up to this point, from the nerve, the action potential coming down the motor nerve, to releasing acetylcholine, to the ligand gates for sodium, to the localized end plate depolarization, to the depolarization wave that spreads across the muscle cell and down the T-tubules. All this is done to release calcium from the sarcoplasmic reticulum so that we can roll the troponin and the tropomyosin out of the way and allow the myosin heads to grab onto the actin and give them a yank. And that's what we do. That's how we get a contraction. Okay. Anyway, so we'll talk more about this on Thursday. Lab today, if you're in lab today, or it doesn't matter what day you're in this week, we're going to start looking at the nervous system in here. So we'll see. It should be fairly interesting. So, because it talks about things we do and why we do them. So, okay. Anybody have any questions before I wrap this up? Okay, then I want to get us out of here. Thank you for being here today.